First, we're going to go over the basic structure of skeletal muscle, and then get into the mechanisms behind the muscle's movement. One whole muscle is made up of multiple components. First is the fascicle, which is comprised of a bundle of myofibers. The myofibers are made up of myofibrils, which in turn are made up of myofilaments. The myofilaments have two filaments. First is the thick filament, comprised mainly of myosin. And second, the thin filaments, made up of actin, troponin, and tropomyosin. These thin and thick filaments form the basic contractual unit known as a sarcomere. The sarcomere has a very orderly and well laid out design. We will begin at the outer border where the Z-disc is. The Z-disc is where the thin filaments attach, moving inward. You can see the I-band. It is the area where there are only thin filaments present. Moving again towards the center, you see that the area begins to transform into a darker region with both thick and thin filaments. This is known as the A-band. Shifting to the right, we come to an area known as the H-band, which is made of thick filaments only that attach to the center of the sarcomere at the M line. Now that a basic overview of muscle tissue structure has been given, let's turn this over to Ricky and go into the mechanisms behind contraction.
In order for a muscle to contract, an action potential must arrive at the synaptic knob of an axon. Once this action potential has traveled the length of the membrane, calcium channels will open, allowing the calcium ions to flood the cell. The calcium ions force the synaptic vesicles down the length of the cell, where they fuse with the cell membrane and release acetylcholine into the synaptic gap. The acetylcholine ions then move across the synaptic gap, where they meet and bond with the acetylcholine receptors on the motor end plate. Once the acetylcholine and receptors bond, sodium and potassium channels open, allowing sodium ions to enter the myofiber, resulting in an increase in ions, which initiates an action potential along the sarcolemma. This newly formed action potential travels down the sarcolemma into T-tubules, stimulating the sarcoplasmic reticulum to release calcium ions from the calsequestrin. Potassium flows out of the sarcolemma into the synaptic gap. These calcium ions flood into the thin filaments and bond to troponin, forming a troponin complex, causing tropomyosin to uncover the active sites on actin molecules. Once the active site is uncovered, high-energy myosin heads leap across the gap, bonding with the active site, forming a cross bridge. The myosin head then pivots back into a low-energy state, releasing ADP, which will then pull thick filaments over thin filaments. These filaments moving over one another causes the sarcomere to contract. Once this contraction is complete, the ATP rebonds with the myosin head, releasing it from the active site on the active molecules. Once this contraction is complete, the ATP rebonds with the myosin head, releasing it from the active site on the actin molecules. This results in the myosin head re-energizing into its high-energy state as ATP is converted into ADP once again. Calcium is then pumped back into the sarcoplasmic reticulum. While this is all taking place, acetylcholine esterase bonds with acetylcholine, breaking it down into its two original components, acetic acid and choline. The choline is then reabsorbed into the synaptic bulb.